Good morning, students. Let us continue with the next topic of our continuing chapter number 13. Next topic is cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in the form of chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named as phosphorylation. If it occurs in the presence of light, then it is known as photophosphorylation. That is synthesis of ATP from ADP and inorganic phosphate in presence of light. When the two photosystems work in series, first PS2 and then PS1, a process is called non-cyclic photophosphorylation. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. Let us see the flow of electron in the figure 13.5. The light when excites the electron from PS1 and PS2, first from PS2 the electrons will be released and will be transferred to electron transport system, then to PS1 and then finally to the another electron acceptor which makes the NADP into NADPH. Both electron acceptors of photosystem 1 and photosystem 2 are entirely different, but finally the electrons released from photosystem 2 goes to photosystem 1 and finally reduces the NADP into NADPH. When only PS1 is functional, the electron is circulated within the photosystem and phosphorylation occurs due to cyclic flow of electrons as seen in the figure 13.6. See how the sunlight excites the electrons from PS1 and received by the electron acceptor, then to the ETS electron transport system and goes back to the PS1. This forms a cyclic process that electrons reach the same destination from where they were released but thereby forming ATP. This is known as cyclic photophosphorylation. Possible location where this could be happening in the stroma lamellae, while the membrane or lamellae of the grana both have PS1 and PS2. The stroma lamellae membranes lack PS2 as well as NADP reductase enzyme. The excited electron does not pass on to NADP but is cycled back to PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not NADPH. Cyclic photophosphorylation occurs when only light of wavelength beyond 680 nanometer are available for excitation. Next is chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism like in respiration in photosynthesis too. ATP synthesis is linked to the development of proton gradient across a membrane. This time these are the membrane of thylakoid. There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of mitochondria. When electrons move through the ETS, we will read this in chapter number 14. Let us understand what causes the proton gradient across the membrane. We need to consider again the processes that take place during the activation of electrons and their transport. Here are some causes of developing the proton gradient across the membrane. First, since the splitting of the water molecule take place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. That is, splitting of water molecule take place inside the thylakoid, lumen of the thylakoid. So proton and hydrogen are produced within the lumen. Second, as electrons move from the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron is located towards the outside of the membrane. It transfers its electrons not to the electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. इलेक्ट्रॉन रिलीज होकर ट्रांसपोर्ट हो उस टाइम सेम टाइम पर हाइड्रोजन कैरियर की तरह एक्ट करेगा. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released to the inner side on the lumen of the membrane. Third, the NADP reductase enzyme is located on the stroma side of the membrane. Along with the electrons that come from the acceptor of PS1, protons are necessary for the reduction of NADP to NADPH. These protons are also removed from the stroma. Hence, within the chloroplast, proton in the stroma decrease in the number, while in the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane, as well as decreases the pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channels of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One is called CF0 embedded in thylakoid membrane and forms transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1. It protrudes out to the outer surface of the thylakoid membrane and faces the stroma. The breakdown of this gradient provides enough energy to cause a conformational change in this CF1 particle of the ATP synthase. It makes several ATP molecules. Here is the ATP synthesis shown through the chemiosmosis. Due to proton gradient, there is higher concentration of proton inside the thylakoid lumen of the thylakoid. This pink colored is the thylakoid membrane. Protons come throughout the membrane through this ATP synthase molecule CF0, due to which the conformational changes occur in CF1 and hence resulting in the bonding of ADP plus inorganic phosphate releasing ATP and the proton comes out. So here are some cytochrome and PS are given to you in order to describe how the proton gradient is increased inside the lumen of the thylakoid. Chemiosmosis requires a membrane, proton pump, proton gradient and ATP synthase. Energy is required to pump protein across the membrane to create a gradient or high concentration of proton within the thylakoid lumen. ATP synthase has a channel that allows diffusion of the proton back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electron, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. Where are the ATP and NADPH used? Once we have studied that during photophosphorylation, ATP and NADPH are formed. Now, the question is where they will be used. We learned that the products of light reaction ATP, NADPH and O2, of these oxygen diffuses out the chloroplast, while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but it depends on the products of light reaction, that is ATP and NADPH. Besides carbon dioxide and water, you may wonder, this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, photosynthesis resumes. Let us now see how the ATP and NADPH are used in biosynthetic phase. We saw earlier that carbon dioxide is combined with H2O to produce CH2O times, which represents a carbohydrate or sugar. It was of interest to scientists to find out how this reaction proceeded, or rather, what was the first product formed when CO2 is taken into reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. Exemplary means very important. The use of radioactive carbon 14 by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway. Hence, it was called Calvin cycle after him. The first product identified was a 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? It have 3 carbon atoms, but glucose have 6 carbon atoms. So this cycle will run twice to fulfill the need. Scientists also tried to know whether all plants have PGA as their first product or whether any other product is formed. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of carbon fixation was again an organic acid, but it consisted of 4 carbons. The acid was oxaloacetic acid, or OAA. Since then, carbon dioxide assimilation during photosynthesis was said to be of 2 main types. Those plants in which first product is C3 acid, that is PGA, are known as to be following C3 pathway, and those in which the first product is a C4 acid are following C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later in this chapter. What is the primary acceptor of carbon dioxide? Let us now ask ourselves a question that was asked by scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting CO2, would have 3 carbons of PGA? The studies very unexpectedly showed that the acceptor molecule was a 5-carbon ketose sugar, ribulose biphosphate. Don't worry, the scientists also took a time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primer acceptor would be a 2-carbon compound before they discovered 5-carbon RUBP. In the further classes, we will be studying C3 pathway, C4 pathway, photorespiration, and the different factors affecting photosynthesis.